My name's Reed Corbett. I'm a faculty at ECU, but I'm a scientist out here at Coastal Studies Institute. I'm very happy to have two of my own colleagues here with us today to give you a presentation on pharmaceuticals and personal care products. And I'll give a little introduction of both of them so you have some idea of a little bit of their background before they came to you. Dr. Jamie DeWitt, she's an associate professor at ECU in the Department of Pharmacology and Toxicology in the Brody School of Medicine. Jamie received her BS degree in biology and environmental science at Michigan State University and a PhD in environmental science and neuroscience at Indiana University. She continued as a postdoc at Indiana and did some time, maybe I shouldn't say it that way, did some time at the EPA. Jamie's work within her lab primarily focuses on the exposure to emerging contaminants in the aquatic environment and how these contaminants can lead to disease such as autism, Alzheimer's disease, after these early life exposure disrupt the developing immune system. Dr. Sid Mitra, who I was thinking about it today as I was putting this together. I've known Sid for, I guess, 18 years now, since we met at Tulane University.
He's a lot older than me, though.
Sid's an associate professor in Department of Geological Sciences, my own home department, at ECU, and also director of the Coastal Resources Management PhD program. Sid received a bachelor's in mechanical engineering at Lehigh University, went on and did a master's in chemistry and environmental science at the New Jersey Institute of Technology, and found his way to marine science at the Virginia Institute of Marine Science just up the road. Sid did a postdoc at Tulane University, which is where I met him many moons ago, and then at the USGS, the Geological Survey out at Menlo Park. Sid's lab focuses primarily on the cycling, fate, and transport of trace organic chemicals in the environment, which you'll hear about today. He does work from black carbon to black carbon and its effect on global carbon cycling, work on contaminant bioavailability, and tracking environmental contaminants to natural waters, again, which is a great lead into what we're going to talk about today, or what they're going to talk about today. And I'll let you take it away.
All right. Thanks, Reed. I want to thank CSI for hosting Jamie and myself, and I want to thank Jamie for agreeing to give this talk with me, tag team. We've never done this before where we're going back and forth like this. So hopefully it'll work out. Hopefully you'll learn something. Hopefully you'll enjoy it as well. The idea of pharmaceuticals and personal care products as emerging pollutants, coastal water. It's not a new one. It's been going on for a while, actually, but we're really starting to find out that it's much more prevalent than we thought it was, and we're also starting to find out that some of the sources are really just sort of our everyday behavior occurrence that leads to some of these sources. So that's me in purple, representing with the purple gloves, doing some field work, and it's very important if you do field work, you represent your school colors. So that's why I'm wearing purple gloves. Okay, so the chemicals we're talking about are the very simple things that you and I take on a regular basis. So I get headaches, I take Advil, or I may take aspirin, I may take Tylenol. On a daily basis, we soap, we shampoo. I have 3 kids who don't do either of those things very often. But you're supposed to shampoo and soap in terms of personal hygiene. And all of those, many of those smell nice. You smell them and they smell nice. Well, the smell is not a natural, in most cases, not a natural substance. It's an organic chemical of some sort. And so these chemicals largely fall under the category of drugs and other things that we take in, as well as care products that we use, consumer products that we use. And so I want to start off by giving everyone a little bit of chemistry, okay? So let's do a little bit of chemistry, and I know that many of you may not have had organic chemistry, but here's how we do it in organic chemistry. We make people, all the students, repeat things after us. So we're going to repeat this, okay? So I want everyone to repeat with me. An organic compound is a compound that has a covalent bond between carbon, hydrogen, sometimes nitrogen, and oxygen. Let's do it one more time. So covalent bond between carbon, hydrogen, sometimes nitrogen, and argon. oxygen. That's a mouthful, isn't it? What if we just simplified that and said they're just stable molecules? Okay? They're stable molecules in the environment. So once they're created, whatever process creates them, they have a stability that allows them to persist, hang out. Okay? In normal circumstances, you're like, oh, what's the big deal? They're hanging out. As we'll find out, that stability leads to some problems in the environment. So here's two chemicals. And I'm not throwing this up just to dazzle anyone with chemistry, but there are two chemicals that are actually quite common, as we'll find out. So this chemical is called 17-beta-estradiol. It's a natural hormone, all right? This chemical is called 17-alpha-estradiol, or ethinyl estradiol. So anyone in this room, anyone, raise your hand and tell me, what is the difference that you see? What are the differences, one of the main differences you see between, if you're not a chemist? I don't want chemists to raise their hands. Chemists know the answer to this. What is the difference? Take a close look at those two molecules. I'm going to ask you to look for, remember you used to play those games where you see pictures side by side, you're asked to find the differences? That's exactly what we're doing now. So what's different right here? You see that right there? That's the ethinyl group. That's what makes this ethinyl estradiol. That slash, each of those slashes is a bond. In the world of chemistry, it's a covalent bond. In the world of chemistry, the more of those bonds you have, one bond, two bonds, three bonds, the harder it becomes to break down. So this molecule is a natural hormone. This molecule was synthesized by a guy named Carl Djerassi, not to be confused with Jurassic Park. This is Carl Djerassi, who's a scientist who was trying to figure out how to regulate ovulation in people. And he was doing this experiment on rats where he would try to dose them with different amounts of this compound, this natural hormone that regulates ovulation. But every time they put this molecule inside of rats, the stomach acids of the rats would break the molecule down because it was not a stable molecule. So he had this aha moment where he said, well, wait a minute. What if I make part of this molecule really stable? Lo and behold, he was the person who created birth control medication. This is birth control medication, ethinyl estradiol, which regulates ovulation in females. Okay? So let's just segue away from that for a second. Let's talk about a study that was done in the 2000s by the U.S. Geological Survey where they looked at pharmaceuticals, hormones, other organic wastewater contaminants in U.S. streams. And you would agree with me that the number of dots on this map, plentiful, right? There's several dots on this map. These are streams and rivers in many places, some upriver of cities, some downriver of cities, but plenty of locations, basically, okay? And here's what they found. This is an overly busy table. Admit it. It's an overly busy table. Too much going on, right? But what we have here is this 17-alpha ethinyl estradiol right here. There's that chemical I just showed you the structure of, okay? And then there's a bunch of stuff on this table, but the thing that I'm hoping you can all see is that here there's a frequency of detection of about 16%. And the concentration, the maximum concentration they detect is 0.3831 micrograms per liter. The median, or sort of the middle of the road concentration, is 0.073 micrograms per liter. 0.831 micrograms per liter. So what does that mean? Let's put things in context. What does a 0.831 microgram per liter mean? Okay, a nanogram per liter is a part per trillion. Part per trillion. So I have one molecule of the birth control medication, a trillion molecules of water down below, whatever unit you choose to measure. If I am looking at something that is a microgram per day, it's in the category of thousands of nanograms per day. If I'm looking at something that's milligram per day, it's in the order of millions of nanograms per day. So a milligram per day. Anyone take any Tylenol or aspirin or anything today? Anything? You took it, right? What? She did it because she knew she had to deal with me for 3 hours. So ibuprofen. If you look at an average bottle of ibuprofen, the dosage on there is about 100 to 250 milligrams. So here we are. That's about how much is in typical medication. When we get to chemicals that are dangerous, the EPA sets water quality standards at these microgram levels here. And then we have even smaller of nanogram levels down here. So that's just sort of a scale to put things in context. Remember, we saw something that was detected in concentrations of microgram per liter, okay? All right, so let's go back to these two chemicals here. Supposedly, human ovulation, natural, human ovulation, synthetic, created with the stability in mind. This study blew away the field of science in 2007 because these authors showed that if I take this drug, which is found at concentrations of micrograms per liter, and I dose them in an experimental lake where it's an experimental lake in Canada where there's no other source of these chemicals, no other pollution, if we dose them, at concentrations of 5 to 6 nanograms per liter. So remember, we're talking about even less than what we detected in the river waters all throughout. It led to feminization of the males, and the males and the females weren't able to reproduce. And if as a species you can't reproduce, this is fathead minnow, if as a species you can't reproduce, what happens to your species? It collapses, right? But the other really neat thing they did in this study was in this experimental lake, they stopped the dosing. They stopped it after 4 years, I think it was. It was a 7-year study. They stopped it after 4 years. After they stopped it, after about year 5, year 6, year 7, the populations bounced back. So there was a reversible effect of stopping the introduction of these chemicals. Okay. So this was the level, roughly, that they were dosing their experimental lake. But see, this is the problem with these pharmaceuticals. What I'm going to talk about today and what Dr. DeWitt is going to talk about today in general, we're going to hop back and forth between the idea that, yes, in the natural waters, when these chemicals make their way out into natural waters, they may exert dangerous effects, but they are pharmaceuticals. They are personal care products. That aspirin has to be taken at 250 milligrams to actually get rid of your headache. So there's pros and cons to these chemicals, which is why there's such a huge problem with trying to figure out what to do when we measure them in the environment. They have a benefit to society. All right, so let's talk a little bit. Let's segue to how do we get these chemicals in the water? Clearly, they're in your medicine cabinet, in some people's medicine cabinet. Your body's involved somewhere. There's wastewater treatment, and then there's local waterway. So let's examine these different sections, okay? Medicine cabinet. So here's a typical medicine cabinet. Here's some allergy medicine, Claritin. Here's some aspirin right there, Bayer aspirin. There's a Aleve right there. A couple of other prescription medications. Some children's toothpaste, which has some other chemicals in it I won't talk about today. And there's some musk right there. So this is an average medicine cabinet. So are these chemicals committing hara-kiri and jumping into the toilet right in front of them randomly? I don't think so. So let's move on to the next box, your body. So that's your body. That's my body on a given day before coffee. But that's your body. Well, it turns out that in your body, depending on the chemical, when you take in a certain amount of substance, and Dr. DeWitt's going to tell you a lot more about this, some of it gets to where it's supposed to go and does what it's supposed to do. But then a large portion of it, in the case of many drugs, go right back out. They go right back out into the wastewater stream. And in the wastewater stream, or if you shower, it's coming out your pores. You know about things coming out your pores. You bathe, you excrete into the toilet. That ends up in the wastewater stream. After it goes into the wastewater stream, it goes somewhere, and it should be treated. But that depends. Sometimes some of the material that comes through your body and gets treated doesn't get broken down in wastewater treatment plants, and I'll show you how in just a minute. Some of that solid is applied to landfills and farm fields as biosolids. It's used as farm material, fertilizer. Some of that liquid is then discharged back into surface waters, and so there's all sorts of different fates. But let's look at this right here. Why isn't this stuff that I'm taking in, it does stink here, but why isn't this stuff being broken down here and poof, going away? Why is it ending up back out in surface water? Let's find out some details. So here is an average city wastewater treatment plant. And treatment is broken down into something we call primary treatment. Most wastewater treatment plants are primary. Secondary treatment, Most wastewater treatment plants have secondary treatment. And then tertiary. Not every wastewater treatment plant has tertiary treatment, but most wastewater treatment plants have primary and secondary. But you know what? Most wastewater treatment plants were designed decades ago. Most of the ones that are in existence in some of the cities that are older were designed decades ago. And decades ago, we didn't have a lot of the chemicals we're coming up with. We're coming up with on the order of 300, 400 chemicals a year, new chemicals for consumer use a year. And so they're just not able to handle a lot of those. And let me explain why. Let me explain the subtleties of why, because I don't want you to go out and blame your wastewater treatment plant operator and say it's their fault. Let me explain what's going on. Okay, all of these chemicals, so lesson number 2 about chemistry. So before we talked about organic chemicals, what are they? What are the organic chemicals? Forget the long definition. What's the one-word definition? Stable. There you go, stable. Okay, so here's another explanation from a chemical perspective. Wastewater treatment plant. Let's say an ideal wastewater treatment plant. Here's primary treatment, the very first part. In general, primary treatment is really sort of a fancy set of words for you let things settle. You let things settle, okay? And let's take some chemicals. So here's a chemical lesson. There are different chemicals in that wastewater stream, that organic chemicals, the pharmaceutical chemicals. There are different ones that have different water solubility. Some dissolve more than others. Some chemicals dissolve more than others, okay? So here's a drug called carbamazepine. Does anyone know what carbamazepine is? It's a drug that's given to people who are epileptic. It's an anti-seizure medication, okay? Here's estradiol. That's that estradiol we talked about. It's a natural hormone, okay? And here's galaxolide. So galaxolide is a chemical that's found in deodorants and musks. It's that nice smell. Quite often it's associated with galaxolide, which is a synthetic perfume-like substance, okay? So let's assume that I have wastewater that's loaded with 100 units of this stuff, 100 units of this stuff. And it's going through the wastewater treatment plant, chugging through, and here's the primary treatment where all that's going to happen is things are going to settle out. Things are going to settle out. Well, what settles out? Well, it turns out that the things that are really soluble, water solubility is much higher here, it's much lower here, things that are really soluble are going to go right through to the other side without falling out. But then things that are less soluble here are going to go through and they're going to settle out. So in the primary treatment, the settling out part, because this chemical is not as soluble, it's just going to drop right out with the solids that are associated in the wastewater treatment plant. And I'm not going to get gross here, folks, but let's face it, wastewater treatment has solids in it, right? Okay? We all know we don't need to say anything more, right? So that material settles back out. And I have fewer of these galaxolides as it comes out of primary treatment. So now let's keep going. Let's go to secondary treatment, where we have some kind of disinfection or something going on, and we want to get rid of bacteria and other things. Remember, wastewater treatment plants weren't designed to treat pharmaceutical chemicals. These were septic tanks. They're designed to make sure that you don't have fecal coliform and other bacteria that are deadly on the other side. That's what they're focusing on. So now we're in secondary treatment, disinfection. And again, if we have the anti-seizure medication, which is not very water soluble, some of it breaks down from the disinfection process, but this, the perfume product, which is very water soluble, breaks down a lot more. There's much less of it. It settles out. It goes away in the secondary treatment process. Now we go to tertiary treatment. So now we've done secondary treatment, and we're only left with about 70 units of this, very little of this. Tertiary treatment, which usually tends to be something called photooxidation or ozonation, will usually break down most of these chemicals, except for some really stubborn ones. Something like sucralose, for example, acesulfame sulfate. Those are both names of artificial sweeteners that are found in a lot of products. Those make it through without being broken down even by tertiary treatment for the most part. So what's happening? Here's what's happening. The big molecule, so the frowny face right here is a pharmaceutical chemical. There's a small one and a big one, okay? Now, To reinforce this point, before we go any further, I need some assistance to help me with the demonstration. So I would like 5 people to come on up. Could I have 5 people? I'm going to call you out. This is what I do in my class. I'd like 5 people to come on up. Ma'am, would you come on up, please? Ma'am, would you come on up? Sir? Come on up. Sir, you back there with the curly hair. Can I ask you to come on up, please? And let's see. Miss, how about you? Come on up. All right. So here's another chemistry lesson. If I tell you that you are a water molecule, you should have 2 elements, H2O, right? So let's pretend that the O, the oxygen, is your head. Let's pretend that the hydrogens are here. The way water bonds to itself is that these little hydrogens like to stick to these oxygens, okay? Now I want you all to cluster together, and I want you to find, your arm needs to find another person's head somehow, and I want you to become a cluster of water molecules. So this is what water is. It's O, H, H, and these H's like to associate with, does anyone have an arm free? No touching anyone below the waist. All right, so here's an arm that's free, and it needs to go to a head. You can go over to this person's head. You can double up right there. That's okay. You can double up, and you can go to their head. All right, now, what I would like you to do is I would like you to think about the fact that you have a big molecule that is not soluble and a small molecule that is soluble. What do I mean by that? Well, you are a cluster of water molecules, and I'm going to ask you to stop from this big molecule dissolving within you. And all dissolving means that you're letting this big molecule within you, okay? You are dissolving it, meaning you are allowing it to come within the water molecules. So I want you to stop this molecule from dissolving in you. Can you do it? I'll be gentle. Don't let this molecule dissolve within you. Okay. It's a fairly straightforward task. I can't get this big molecule to dissolve within these water molecules, right? I want you to do the same thing with this. This is a marble. I want you to stop this from dissolving within you. Not very successful, right? So thank you very much. Can we give them a round of applause, please? So the principle, one of the principles, and there are other factors that affect it, is that the small molecules work their way the wastewater treatment. They stay dissolved. They work their way in between. They don't get stuck on anything. The big ones can't make their way through water molecules, so they associate with whatever else they can find. Surfaces, such as solid waste inside of the solid waste stream. So let's go back. Particles in the waste stream, that's, whoops, oh, that was me. Go back to
play over to the right.
La, 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 la. Thank you. So the big ones, the bigger molecules, tend to associate with particles. They settle out. They're less likely to work their way through your wastewater stream. Okay? So now, I've told you what types of chemicals we're talking about. I've told you how they sort of make it through. Okay? And I want Dr. DeWitt to now come up and tell you about what happens when these chemicals end up in waters. And then I'm going to come back, if you want me to, I'm going to come back and tell you about North Carolina and coastal areas and what we know about some of these chemicals and their survival in coastal waters. Okay?
Thank you. Well, I'm not going to make you do a demo. I'm not going to embarrass you and make you stand up here. And now, Dr. Mitra told me that I needed to get a better picture. He said I needed a field picture. But this is from a field site. I'm at a laboratory in Las Vegas. That was my joy of Las Vegas, sitting in a laboratory. And actually, that was a pretty big laboratory. I spent most of my time in a 10 by 10 room. So I'm going to talk to you a little bit about how we try to figure out what these compounds do once they get into your body. So Dr. Mitra has told us how they get into the environment, but he's also told you that these are naturally produced by our bodies. We take these to make ourselves feel better, We put these on our bodies to smell better. So why are we even concerned about them at all in the environment? Before we do that, I'm going to tell you a little bit about my field, which is toxicology. So a lot of people think that toxicologists are the mad scientists. I think when we're displayed on television, we're the evil scientists in the corner developing these hazardous agents that we're going to send in the mail. Well, that's really actually not true. I think the chemists do that. So when you think of a mad scientist, think of Dr. Mitra. When you think of somebody whose agenda is to understand how hazardous agents impact us and how we can try to reduce those impacts, I want you to think of me. So we are not really angry, mad scientists. We might be angry about some things, but we're not mad, we're not crazy. We really try to understand the health effects of toxicants or pollutants that go into the environment. As an environmental toxicologist, I focus on contaminants that end up in the environment. I also study the developing immune system and the developing nervous system to understand how early life exposure to these agents impacts us as we grow and develop and age. So as Dr. Mitra said, some of the disorders I study are autism, others I study are Alzheimer's disease. And I believe these actually have some commonalities in terms of early life changes that lead to these later life diseases. So when I'm talking about these compounds, I'm trying to figure out what they might do to our bodies when we ingest them at times when we shouldn't or at concentrations that we shouldn't. Now, there are all sorts of different contaminants in the environment. The ones that I study are these PPCPs, pharmaceutical and personal care product pollutants, but I also study PFASs. These are called per and polyfluoroalkyl substances. The chemists name them, I don't name them. I just try to say the names properly so that I don't sound like too much of a fool. But in our environment, we have a lot of different agents that we use every day. Many of these agents are important in terms of protecting our health. PBDEs, for example, are flame retardants. They are put into furniture and clothing to reduce the possibility for flame during a fire. Nanomaterials are in our makeups to make our faces nice and shine-free. Chlorinated compounds are in pesticides that help to improve yields of our different farm products. And heavy metals are in the things that we sit on, in the buildings that we stand in. So these compounds are vitally important to our livelihoods. They're important for our health, they're important for our well-being, but problems arise when they get into the environment and we get exposed to them through ways that we shouldn't get exposed. So as an environmental toxicologist, I try to understand sort of the whole exposure profile, so how the chemicals get into the body, what they do to our body, and then what we can do about it to reduce the exposure and reduce the potential health effect. But we have to understand the health effect first before we can start to say, okay, what do we need to do? Now, these emerging contaminants are contaminants, as Dr. Mitra said, for which we have few to no regulations. So these are contaminants that we know are in the environment, but we're not regulating them. So we're getting exposed to them, and there is no way to stop our exposure, mostly because we don't know what they do to us. We don't know the potential hazards. We can't quantify the risk, so we can't regulate them. Well, yeah, you're not going to put those into the environment. You're not allowed. How do you think that would fly? Most companies that rely on these compounds for their livelihoods would say, well, they're not hazardous, so I'm going to continue to put them in the environment. This is a photograph of Dr. Mark Striner. He's an analytical chemist, like Dr. Mitra, from the United States Environmental Protection Agency, and he's out sampling along the Cape Fear River. He samples and he collects water to look at the concentration and the type of these per and polyfluoroalkyl acids that I study. So he collects samples and then he tells me what he's found. Once I know what he's found, then I can start to make some decisions about what agents I should study in my laboratory. So I just told you why we have compounds in the environment that are not regulated, because we don't know the hazards, we don't know the risks. One of the functions of the United States Environmental Protection Agency through the Safe Water Drinking Act is to evaluate our water and to determine what compounds are in the water and what compounds need to be studied. How many synthetic chemicals do you think exist in the world? What's that?
Millions.
Oh gosh, I hope not. There are probably about 80,000 synthetic chemicals. Many of these, we don't know the hazards. We haven't studied them. So one of the duties of the United States Environmental Protection Agency is to come up with an assessment of their risk to determine if they need to be regulated. And that's where people like me come in. I study their toxicological effects and publish papers that indicate, yeah, this stuff is pretty bad, or, you know, at concentrations in the environment, it's not so bad. So the list that the EPA keeps that tells us about contaminants that are in the environment, but for which we have very little data, is called the candidate contaminant list. You can look it up. We're on the 4th list now. It's called the CCL4. You can find it through the US EPA's website. And it lists all of the agents that are known to occur in water. It's a fairly hefty list, and it contains pesticides, it contains metals, it contains industrial compounds, and it contains some of these pharmaceutical and personal care product pollutants. So we do have stuff in our water that isn't regulated. So this water that I'm drinking has chemicals in it that are not regulated. I don't know, to me that's a little bit concerning, but maybe these chemicals don't do anything to our bodies. How do we know? So what do we do to understand these chemicals in our bodies? Well, we really don't know very much about these emerging contaminants. That's why they're emerging contaminants. And we kind of think what we need to know about them from a toxicological perspective, but in reality, there's probably a lot that we don't know. And one of the reasons is that most of these chemicals are found in mixtures. There's not just one chemical floating around. There's thousands of chemicals floating around. And for the most part, we really don't know how these mixtures interact in our bodies. I guess I'm the doom and gloom portion of the talk. Yeah. So what types of questions should we ask to try to understand what these compounds do? Well, we need to know where they come from. How do they get into the environment? What concentrations are they in the water? Do they get metabolized by bacteria? Where do they go? Do they stay just in groundwater? Do they go to surface water? Do they go to the estuaries? Do they get into the marine environment? Well, do they move easily? As Dr. Mitra said, are they water soluble? Do they bioaccumulate, which means do they concentrate in biological organisms? And then what about in us? Okay, if we're drinking them, do they get into our bodies? Do they stay into their bodies? Where do they go? How do they get out of our bodies? There's a lot of questions that we have to ask to start to understand how these compounds work in the environment. And one approach that we can take is we can start looking at wildlife. We can use wildlife as the canaries in the coal mine to understand some of the potential effects of these compounds. So we might do field observations in wildlife, or we might collect some samples from wildlife and look at their cells. I like to call this is the sexy side of toxicology, because look at what these toxicologists get to do. My friend Jennifer lives in Hawaii. She gets to collect samples from sea turtles. She gets to ride around on their backs when they're laying eggs and collect skin samples. So she lives in Hawaii and she maintains a sea turtle database, a tissue bank, so that she can understand the effects of contaminants in sea turtles. We also have folks who get to go out and sample dolphins. Supposedly, I get to go on a dolphin sampling trip someday. But on these dolphin sampling trips, we can collect samples from the dolphins to understand not only what might be in their tissues, but whether or not they're healthy. So we can do assays with the samples that we collect. We can collect blubber, we can collect blood, and we can ask questions about what these contaminants might be doing to their health. And we can also do the same thing with wild birds. So we can go out and we can collect samples from wildlife to understand what might be happening to wildlife and how much of these contaminants are in their bodies. But it's really cool, but it doesn't really tell us the whole story, because we're part of the ecosystem too, and we probably want to know, well, what's it doing to us? If it's doing something hazardous or harmful to a dolphin, is that applicable to me? I don't live my entire life in water, so what is the applicability of the dolphin to me? So this is where we start to move into other types of model systems that have better applicability to humans, but that still are closely related to wildlife so that we can understand those two relationships. So a lot of methods we use in toxicology are vertebrate animal models, such as mice. Some people use cute little bunny rabbits. I don't use bunny rabbits. But we use these vertebrate animals and their cells to understand what contaminants in the environment might be doing to us. And then we relate those samples to wildlife so that we have an entire picture of what's going on in terms of exposure. And so when we do this, we have a slightly better approach. So we can make observations in wildlife, and we can look at wildlife cells. We can compare those to laboratory animal models, and then we can do studies with model systems as well. So we can also eventually put this into a computer model to make predictions about potential adverse outcomes, and then not take any samples at all. So as Dr. Mitra mentioned, this is a pretty complex problem, and understanding exposure means understanding all of the different types of compounds that could get into the environment. So we have livestock that are exposed to pharmaceuticals. They're exposed to various antibiotics and other drugs to keep them healthy, so they're contributing to the wastewater stream as well. Hospitals have effluents. A lot of people at hospitals take a lot of medications. That all goes to the wastewater system. We have contaminants at home. We have manufacturing. All of these different sources contribute to what's in surface waters. So if we just ask one question about what do people use at home, we're only looking at a very, very small piece of the puzzle. So the idea behind this slide is to emphasize that this is a pretty complex problem. It's not an easy question to address. And these contaminants have likely been in the water for decades. So are we experiencing any adverse health outcomes? Anybody have any hair growth that they don't want? Any odd health outcomes? I don't know, maybe. And as Dr. Mitra indicated, improper disposal and regular disposal is one of the ways that these compounds get into the environment. But if you remember from this slide, it is more complex than just improperly disposing of medications. Okay, so one of the questions that one of my students had during her seminar was, well, none of this stuff matters because all of these agents have been approved by the Food and Drug Administration. They are all safe for us to use, and the concentrations in the environment are way, way lower than what is even efficacious or therapeutic. So these compounds in the environment are at such low concentrations that it doesn't matter. So why should we care? So what? I took ibuprofen today, so I'm going to get rid of some of it before I hit the road. So what? Why should we care? This is a question that you can answer. This is my level of audience participation. So why should we care? Most of these are approved to be safe. We can ingest them. We can put them on our bodies. We can put them into our bodies. So why should we care? You are all here tonight, I'm presuming, because you care. Are you just curious? Scientific curiosity? Well, we should care because they can induce health effects. Dr. Mitra mentioned the study of the fish population that collapsed. So these male fish were exposed to estrogens. Those estrogens increase concentrations of vitellogenin. This is a protein that female fish make in response to estrogen. It's a protein produced by the liver that helps them to make egg yolk. Males shouldn't be making vitellogenin. So the fact that males had vitellogenin says that they were exposed to an estrogen. And so that indicates that these males were feminized because of the agent in the environment at concentrations that are extremely low, lower than concentrations that we would take for birth control. These fish were immersed in this compound, and these fish were likely immersed in the compound throughout a major portion of their life cycle. So effects in wildlife can inform us about potential effects in humans. And these are a collection of various papers that indicate we're starting to understand that low concentrations of these agents in the water can lead to adverse health outcomes, mostly in wildlife. Now, adverse health compounds is just one of the questions that we have to address. Mostly we want to know, how do we manage the risks? Once we identify them, how do we manage them? How do we reduce these contaminants into the environment? But remember, from that complex picture, we have to understand how they get into the environment, what allows them to move around in the environment, so their chemicals of their chemical characteristics, how they induce toxicity, if indeed they induce it, and maybe other environmental factors that might contribute to our understanding of adverse health outcomes. Now, there are many sources of uncertainty associated with these chemicals. Low concentrations. Can these low concentrations produce effects? And do we have to look for different types of effects? It used to be back in the days of Rachel Carson in the 1960s when she wrote Silent Spring, one of the main health effects was cancer and death. Okay, those are two pretty scary outcomes. But now we focus on sublethal effects. That is, effects of compounds that are not strong enough to induce death. And these are usually outcomes that are associated with the nervous system, the endocrine system, the immune system, the three main controlling systems in our body. I mentioned the issue of mixtures, and then there's the issue of metabolites. Do these compounds get broken down into others in the environment? Well, I need a chemist to tell me what these compounds are. And the impact of life stage. So these are all different types of questions that we have to ask to start to design appropriate toxicological studies to get a handle on what exposure to these agents might do to our health. Now, in terms of what we've observed in wildlife, we've seen antibiotic resistance, endocrine disruption, immune dysfunction, nervous system dysfunction, impacts on specific organs, and developmental effects. And I've highlighted these because these are some of the endpoints that we're studying in my lab. And the goal is if we decrease uncertainty, we also decrease risk. So the more uncertainty we have, the more potential risk we have, because we don't know whether or not exposure is going to induce health effects. So in terms of who is most at risk, it's usually going to be developing organisms. Developing organisms are undergoing a lot of cell division. Anytime there's a lot of cell division, there's an increased potential for something to go wrong. Developing embryos don't even have their organs formed yet. So if an unborn child is exposed to one of these agents, they could have malformations in one of their organs when they're born, anywhere from severe to subtle. The elderly, sorry Sid, the elderly become inefficient. He joked about being old. They become less efficient at metabolizing and excreting, and so the elderly can be at increased risk to exposure. And then those with compromised health, especially those with immune systems that are compromised. If you have a compromised immune system, then you're more susceptible to disease and infection. So in my lab, we're studying three main compounds. Fluoxetine, anybody know the trade name for that? You don't count. Prozac, okay, it's a selective serotonin reuptake inhibitor. It acts on the nervous system. We're studying fenofibrate. This is a drug that many people will take to help to lower cholesterol. And norethistérone is a progestin. It's also involved in birth control medication. So our approach to understand effects on the endocrine system, the immune system, and the developing system is to use these different agents in a chicken model. So we're putting these agents into chicken eggs and looking at the effects of these agents on a developing chicken. And we're especially interested in what's known as the stress axis. So the stress axis is the axis from the hypothalamus, which is in your brain, to your anterior pituitary, which also is in your brain, to your adrenal gland. So the main compound in the stress axis is called corticosterone in birds. In humans, it's cortisol. It's your stress hormone. So I probably have high levels of cortisol right now because I'm standing up in front of you talking. So I'm getting stressed out. But that's a good thing because it makes me a little more hyper. It makes me care about talking to all of you. If I didn't care, if I didn't have any stress, I'd go, oh, yeah, so this is what we're doing. Yeah, we're putting stuff into chickens. Yeah, and Dr. Mitra is going to talk a little bit more. But if I had too much stress, I could have a panic attack and faint. So ideally, our stress or our cortisol should be at a level that keeps us ready for that fight or flight. So it keeps us prepared, it keeps us energized. Too much can be bad, too little can be bad. So we're asking questions about how developmental exposure to these compounds impacts stress. And we found that there are some small changes. So this is a graph of these different drugs, and this is something called the glucocorticoid receptor alpha. It is the receptor in your body that cortisol binds to. And we see some changes in it when we compare the drugs to embryos that have not been injected. We're also looking at how the DNA responds. So we're asking questions about changes to DNA that are related back to the glucocorticoid receptor that tell us how the organism responds to stress. And the idea behind doing this is, as I said, if an organism doesn't respond appropriately to stress in its environment, then they're not going to be able to fight off infection. They're not going to be able to reproduce. They're not going to be able to escape predators. So we're asking specifically, how do these compounds impact an organism's ability to respond appropriately to stressors in their environment? So in terms of what else we're going to do, we're interested in looking at immune function as well. So once we address questions about corticosterone, we're going to ask whether or not the organisms can respond to an immune challenge. Can they fight off infection? Because we're not interested in whether or not they're just going to keel over and die. We're interested in these sublethal effects. And that's all I have to say right now about toxicology. Dr. Mitra will go ahead and give you the happy part of the talk. Yes, sir.
Do you have any idea what the relative amount, contribution of the various sources of these pharma PPCPs in water?
Dr. Mitra is going to talk a little bit about some of the sources here in the coastal plain in eastern North Carolina. I don't want to steal his thunder.
Okay, so so I want to go back to North Carolina and coastal waters in general and talk a little bit more. So we started off, like I said, giving you some background about the chemicals. Jamie told you a little bit about what they can do, how they study what they can do, and so I want to go back and bring it back home to what some of the issues are with a coastal place. So the reality of the situation is most places in coastal areas, and many areas around these counties here on the coast, most of these places really they don't use primary, tertiary, secondary, tertiary drinking water treatment. They use septic tanks, okay? Septic tanks tend to work in the following manner. Basically, things come in from the house into the tank, solids settle out. Remember that primary treatment, we talked about settling out? Same thing going on right here. Things are settling out. There's a little bit there are a bunch of bacteria in here. The bacteria are breaking things down. There's an outlet pipe that then leaches out into a drain field. And that's how septic tanks work. Most of the U.S., or a portion of the U.S. population is on these types of decentralized wastewater treatments, about 25% of the U.S. But when you get to coastal states like North Carolina, South Carolina, Alabama, West Virginia, the number goes up. In coastal systems in general, many places tend to exclusively rely on septic tanks. In North Carolina, tens of thousands of new permits are issued each year for on-site treatment systems, septic tanks in people's houses, okay? And the way it works is once things leave the tank, they go to a drain field, and you should have sorption of some of these materials, these pharmaceutical chemicals. They should stick onto the soils. Remember, what did we say when we showed the 3 different types of chemicals going through the wastewater treatment plant? It depends, right? The ones that are smaller tend to be more water soluble. They don't get stuck. They work their way through. And if you have groundwater, they might find their way into groundwater. So in our world, in the world of scientists, we have something called peer review. And peer review essentially means, it's a good thing. It means that other scientists have reviewed your work. And so I was involved with the Albemarle-Pamlico National Estuarine Partnership, it's a mouthful, and actually trying to find out how many studies there have been on pharmaceuticals in North Carolina, let alone in the coast. In all of North Carolina, there is a total of 20 studies that have been done in the entire state. And of those 20 studies, the majority of them are actually in the western part of the state, in the mountain streams. There are a few here, and our group has contributed to a few of these. And a lot of them are not peer-reviewed. And when I say it's not peer-reviewed, it's not necessarily a bad thing, but it's you might read something in a magazine, for example. Magazine doesn't necessarily go through a peer review process. There's no other scientist saying, okay, these data are good. It might be someone's high school project. It might be something else, but it hasn't gone through a process of vetting and some type of authority reviewing it. So that's about the total number of, I did this back in 2015, so it may be a little outdated, but very few studies. So here's what our group has done in terms of some. We've published 2 papers on pharmaceuticals in North Carolina. Here's Pitt County, and we looked at some drain fields down gradient of a septic system in eastern North Carolina, and down gradient across the tank, across in the tank or down gradient of the tank. Some of the chemicals we found in general were, of all the samples, we found DEET in about 78% of them, caffeine in about a third of them, a little bit more than a third, homosalate, which is sunscreen, which was found in about 20% of them, ibuprofen in about 40% of them. And we have more data on these, but I don't want to show you guys data. I want to talk about sort of general trends. So we find these down gradient in streams. We find some of these in headwater streams as well that are way down gradient of the actual tanks, okay? So, and sir, I'll get to your question in just a second. So I want to sort of end it, before we get to questions, I want to end it on, this isn't supposed to be a panic talk, so I want everyone to understand, this is where we're at. This is where we're at. We have septic tanks. Some places we have wastewater treatment plant systems. We have these chemicals that aren't regulated. We generate a lot of them because of processes where we need these chemicals for our benefit. This is where we're at. So what should you do? Should you be concerned? Maybe. But first thing is, in general, for any type of chemical loading, we say, let's reduce it. Well, use less. If you're concerned about a consumer, about the general effect, use less. That's one thing you can do as a consumer. If there are certain consumer products that are magical, they say antibacterial, scratch your head. Say, what does that mean, antibacterial? Well, if it's bad for bacteria, what is it that it's doing in the environment? There's a lot of good bacteria in the environment, and so, ask yourself questions. Simplest thing, if you are concerned about the water you are drinking, if you're concerned about the water, and I'm concerned about the water I'm drinking, and I've tested the water that I'm drinking, but I still feel that if you get yourself a $30 kitchen water filter of some brand, you can name the brand, whatever it is. They all work equally well for the most part. Your refrigerator, a lot of modern refrigerators have nice carbon-based filters on them. Those carbon-based filters filter out a lot of these chemicals as well. Get yourself one of those handheld filtering devices. Use your refrigerator's filter. Change the filter frequently. You'll be fine in terms of exposure to many of these chemicals. Most drinking water treatment plants tend to do a pretty good job. Greenville, where I live, they do a really good job. I've tested their water for pharmaceuticals before. Obviously, don't flush unused medications down the toilet. That's a big no-no, because just the sheer amount, use less. If you flush unused medication, you're taking like 100% and throwing it right down into the wastewater stream, okay? So not that much information in coastal North Carolina. Why? The chemicals aren't regulated. The chemicals we tend to study, usually that government agencies pay us to study, are the ones that are on a list that say, yeah, these are bad things. Benzene, toluene, xylene that come from gasoline and hazardous waste sites. These chemicals, not so much. So now, sir, you had a question, and your question, well, let me just wrap up. I want to acknowledge some people. I have students in both the university I was at before at Binghamton as well as ECU that have helped with this research. Colleagues, Charlie Humphrey, Mike O'Driscoll, and then Dr. DeWitt has some friends and collaborators, or students and collaborators that have helped her with this project as well. And we've gotten funding from a variety of sources to try to get some of these data and present some of these results to you. So I want to acknowledge all of them. But now I want to open it up for questions, both personal, random, whatever. And I want to start with yours since you asked yours earlier. And if you could repeat the question, please, into the microphone, it would be great.
What I'm interested in learning is how much you know about the relative sources of these contaminants. I mean, one of the things that you might do to try to combat it in a non-chemical way is by knowing where most of them are coming from, whether they're the home, whether they're industry, whether they're runoff. I mean, I'm involved in a watershed up in Massachusetts, and we're starting to think about looking at things like this, and any help that you might give on where you think they might be coming from in your coastal environment might be applicable to our coastal environment.
The good news for you, sir, is actually Massachusetts is far ahead of the game. Massachusetts and California are the two states that are really light years ahead in terms of finding the sources where they're coming from. Where in Massachusetts, may I ask? Plymouth, okay. So Massachusetts has an organization called the Silent Spring. Yes. You should Laurel Schrader, yeah. She's wonderful. She's actually done a lot of work on groundwater transport of these pollutants. The answer to your question is that it's complicated and it depends on where you're at. In North Carolina, we have a lot more agriculture than a state like Massachusetts, for example. So agriculture is a large source because agriculture tends to have these chemicals that are introduced into organisms for their growth. So there are growth hormones that are involved that are excreted. It's not the only source. There's antibiotics that are used. It's not the only source. Humans that are on septic systems, depending on the pharmaceuticals they take in, they make it through septic systems. Many people use artificial sweeteners, and like I said, those are some of the most stable chemicals that are out there. Are they all harmful? We just don't know. But here's the thing I would say about harm. Jamie mentioned that in terms of harm, the two big things we should look at are cancer and death, okay? One of the things we're starting to find out now is that there are populations, human populations, that tend to have statistically relevant elevated responses of certain things like nervous system disorders or high blood pressure. And so it's complicated. I say it's complicated not as a cop-out, but because there's so many chemicals and there's so many effects, and a human is exposed to so many things throughout their life that you can't say, that's it. It's that septic tank right there. It's a very, very difficult thing to do. But at the same time, the data don't lie. There's a reason why there's a higher cluster of people showing susceptibility to infection that happen to be an area that is down gradient from an agricultural facility, possibly a CAFO. It depends. So agriculture is definitely involved. Septic tanks and domestic and on-site treatment facilities are involved. A lot of it is the type of groundwater that you have, whether or not it ends up in a stream. A lot of the aquifer and the soils that you have. A lot of it depends on the types of buffer zones you have next to streams and rivers. Buffer zones are good because buffer strips tend to absorb a lot of these chemicals, and so it's a very complicated answer. And in North Carolina, I can't pretend to know what I can attribute to which of those sources. But definitely agriculture, definitely on-site treatment, and in some cases, wastewater treatment plants don't necessarily use tertiary treatment. Some things can tend to come out. Some of these chemicals can tend to come out as well. But they're not regulated, so it doesn't matter if they come out. Reed.
Are hospitals required to monitor any of this that's leaving their
The
drain?
effluent?
Hospitals, yeah. What's actually leaving the hospitals, given that what we're talking about here and knowing that it's making the environment, it seems like hospitals could be a hotspot.
Hospitals can be a hotspot, and hospitals are no different from chemical plants in that they have to obtain permits for discharge of, they're permitted discharge facilities, basically. And from the wastewater treatment plant's perspective, their attitude usually is if it's not regulated, and as long as they know when it's being discharged, they're better able to control for it in the sense that they can spend more time on an ozonation process if they know that X, Y, and Z hospitals are going to be discharging on such and such day. So most industrial facilities that do discharge into municipal wastewater or into wastewater treatment plants tend to be permitted to do so.
But permitted for certain compounds, not permitted in general. We're
Right.
talking about a National Pollutant Discharge Elimination System permit, which is a federal permit that's managed at the state level. And so you have to know what to permit It's
Right.
not just everything, it's certain lists of compounds.
Right. So for example, some of the bigger pharmaceutical firms we have, the chemical firms in the Greenville area, they have to disclose that here are the things that we discharge high levels of on such and such day, et cetera. So yeah, they do have to say what they're discharging.
Well, and there are different types of permits too. The National Pollutant Discharge Elimination System permit allows companies to legally discharge certain compounds. But through the Toxic Substances Control Act, Community Right to Know, we also can get a list of what chemicals are emitted by companies into the environment. So you can actually look it up. You can go to the EPA's website and you can look up your zip code and you can find a list of all of the chemicals that have been emitted into the environment, what they are and how much there are. It's through the Toxic Release Inventory under TSCA and the Community Right to Know Act. So as a community member, you have the right to know about the contaminants that are in your community. But not every single contaminant is listed because not all are regarded as potentially hazardous. So compounds don't become listed until we identify their hazards. And we usually don't identify their hazards until after we find out that they're already in the environment. And then we go, oh shoot, they're in the environment. Guess we better try to figure out what they're doing.
So how should we dispose of our pharmaceuticals at home?
So if you have medicines at home that you need to dispose of, most pharmacies actually take them back. In fact, almost all the major pharmacies will actually take unused medications. And what they do with them to dispose of them is they usually they send them to incinerator facilities. So that's what's usually done.
But there are also opportunities through a lot of local police departments will do Operation Medicine Chest.
Right.
You can package up all of your unused pharmaceuticals into a Ziploc bag, drop it off, hand it to the police officer, no questions asked. They take it and dispose of it properly.
Yeah. This gentleman had a question over here.
There's a condition called temporary global amnesia that seems to be a lot more common than I would have guessed. From 50 years ago, I don't think it even existed. Is there something in the chemical effluent that might cause something like this?
This one's all you, Jamie.
I don't even know where to start. I ask myself that often because before I started studying these per and polyfluoroalkyl acids, I studied dioxins and polychlorinated biphenyls, which are stable, persistent, organic chemicals that bioaccumulate in the environment, bioaccumulate in our bodies. And so we have all sorts of regulations on dioxins and PCBs. And now I'm working with compounds that are stable, organic, persistent. We think they bioaccumulate, but we didn't regulate proactively. We don't follow the precautionary principle. We always seem to address the problems, I don't want to say when it's too late, but when there's already global environmental pollution. And all I have to say to that is vote in your local and state and national elections.
So one of the things so let's turn this around now. There's obviously consumer benefits which lead to economic benefits for the companies that make some of these chemicals, right? And they're not, they're just trying to make a chemical that helps a human being deal with something, okay? So you then come along as a scientist and you say, you know what? Company X or company Y, that stuff you put out in the environment, it's bad. It's bad. It's causing X, Y, and Z to happen, and amongst organisms, some crazy scientists, they figured this out. I don't know. So what's a company's reaction? Well, first thing the company usually does is, okay, how bad is the reaction? Is it a regulated chemical in the sense of how dangerous is it? If it's not regulated, all right, what can we do? Are we getting public pressure to deal with this chemical? All right, so depending on that answer, the chemists in that chemical will what usually try to do is they'll say, okay, but we need something that does that good thing that our chemical X did. We need it to keep the plastic from melting in your dishwasher, so we got to put something on that plastic. What can we design that is very close to that chemical? So they'll design something. a chemical. They'll synthesize it. They'll say, okay, let's make something now that looks very much like the other, does the thing that the other chemical did, but it doesn't have this property of killing 10,000 babies every 1 million, or causing this problem or whatever, okay? Great. But something you have to keep in mind, and this is how I think of some of these issues, is that that chemical still had to do something that the other chemical did. So there's some similarity in property. You can't get around that. I mean, there's some similarity in chemical property. And so some of the toxic effects and the relevant effects to the environment are going to carry over. It may not be as much. It may not be a chemical that's yet regulated, but it's still behaving similarly. And I bring this up because there's something that's going on right now about plasticizers. There's all sorts of research about plasticizers that are showing that plasticizers, things that are used to coat plastics, are dangerous. And now they're saying there are plastics that are BPA free, bisphenol A free, or phthalate free. Well, whatever you put on that plastic still has to do something, and chances are, just because we haven't looked at that chemical doesn't mean that there's not some detrimental effect from it. And so it's a give and take. We go back, but as a consumer, you have a right to ask what's in this substance. You have a right to know what's in my neighborhood. You have a right to know what's happening to me as a result of this. These are all things you have a right to know, and you should be curious about it. Yes, sir.
This is not intended to be an uncomfortable question for either of you.
This one's yours, Jamie.
I'm trying to think of a way to answer it. But if the public sentiment were that all of your references to regulation is something we're not interested in fact, we're overregulated. And your list on the EPA list of possible things, maybe we don't even want the EPA to have a website that would give us that information. I can imagine that would have a chilling effect on you and your colleagues in this country. My question is this work being done in other parts of the world besides the United States, so that a chilling effect here would not necessarily mean a halt to the intellectual pursuit of the public health?
Yeah, it's a very timely question. I think we're both going to say the same thing. Europe is light years ahead. They're light years ahead. Canada is light years ahead on some of this research. In fact, a lot of the studies that keep coming out that show, and part of the reason is Europe is very concerned because they have smaller borders, and so they need to know where things are coming from, where they're going, whether it's atmosphere or water or groundwater, et cetera. And so yes, the science is going to go on. Australia, New Zealand, a lot of places are doing. I mean, most of these papers are coming from all over the place. China, India, they're doing it as well. That said, a lot of countries, countries such as China and India, they're at a place where we were as a country 60, 70 years ago, where they're finding out that a lot of these industrial pollutants and agricultural pollutants are having some serious health effects on their population, despite the science that we know to exist throughout the world. And so, both experiments are going on. The scientific experiments are going on. The experiments where people are accidentally exposed, that's also unfortunately happening in all parts of the world.
Yeah, the same thing. The European Union is much more stringent in terms of chemical protection than the United States. So we, as Sid said, we're behind other countries in terms of putting into action some of the science that we're developing to protect ourselves from the potential exposure and the potential hazards that might arise from that exposure. Now that being said, I'm drinking out of a plastic bottle. I do tend to shower, so I use these products. Yeah, I tend to shower. I use these products. I try to make decisions that are informed. I don't tend to drink out of plastic bottles. I usually carry my own bottle. Whether or not we're over-regulated, I think really depends on your perspective. If you're if you're working for a large multinational chemical industry, the challenge is meeting regulatory requirements of multiple countries, and it can be very overwhelming. I have many good friends who work for companies that make pesticides and work for companies that make many of the synthetic chemicals that we use and we depend on. And the scientists who work for those companies are very concerned with being good environmental stewards. And so really what we have to do is balance the public good with the potential public cost. And I think that's really the challenge in regulation. It's not, we're banning this chemical because it's horrible. It's what is the acceptable level in the environment that produces the lowest risk, and what data do we need to ensure that we're reaching that level of uncertainty that's associated with low risk. So I don't think it's an issue of banning particular compounds. It's like having a breed-specific ordinance against pit bulls. You're not really eradicating the problem, you're just displacing it. And I think when we eliminate certain compounds, we're going to come up with alternatives because we still need the product, we need the use. We need pesticides. We have 7 billion people to feed. We need to ensure that our farms produce the food that we need to survive. We need gear to keep us dry and warm when we're out in the environment. So the question is, what is the balance? And I don't think any country has really come up with the best way to balance the public good with the potential public cost. If you have a solution, you should run for president.
I'm disappointed in a way to hear how not at the head of the curve the United States is, but I am satisfied and pleased to hear that other countries in the world may not be falling to the same chilling effect on science that we currently appear to be.
In a way, though, even though some of the other countries are ahead of the curve in terms of numbers and types and breadth of regulation, they also don't do the same depth of testing that we do. So, for example, in the European Union, nobody works with vertebrate animals much anymore. They have so many restrictions on vertebrate animal work that they can't do the type of work that they need to do to understand some of the questions that we need to address. We have great computer models, we have great cell lines, but they in no way recapitulate the intricacy of a developing organism. And so we have to understand some of these processes in living, breathing organisms, and unfortunately we can't do that in humans. And so the European Union depends on us to provide those data. And if we can't provide those data, then other countries with fewer regulations on humane treatment of animals will be providing those data. So they might be ahead of the curve on us in terms of implementing regulation, but in terms of generating the scientific evidence, I still like to think that the United States is at the top in terms of the depth and quality and quantity of our science. I think we have among the best scientists in the world. Now, if somebody's watching from another country, I've probably just insulted a bunch of people, but we have some of the best systems for understanding scientific problems in the world.
Thank you very much.
Do we have any questions from people typing in or anything? Okay, all right.
Hi. I've actually done some of this research on fish in Illinois, and where our landscape is about 3/4 agriculture. We've had lots of noticeable population trends of fish, and cricket frogs was a big one that's been published. Even prostate cancer in farmers in the area. I was wondering if there were any of these trends in wildlife in North Carolina anywhere that could be indicative of.
So some of the western side of North Carolina studies, Greg Cope has done a lot of work on freshwater bivalves.
He's at North Carolina State.
He's shown that, and I don't want to misstate his work, but he's shown a lot of detrimental effects to immune systems using freshwater bivalves, but that's as far as I want to go and say what he has shown. But yes, there has been some work on organisms.
Well, yeah, there's a professor at UNC Wilmington named Suzanne Brander. She's done a lot of work with looking at blue crabs. in the estuaries and looking at endocrine disrupting effects. So looking at hormonal changes in blue crabs and how that affects their reproduction and their numbers and their availability to capture for delicious food.
And that's exactly what I worked on as well. So
Yeah.
I was just curious because of how heavily we depend on the fishing industry here.
And there's also some folks out west who are looking at some of the effects of these compounds at the level of fish. So Shea Tuberty at App State has been looking at some of the effects of these compounds in fish species as well, looking mostly at numbers and I think distributions, but he has, I think, started looking at some molecular level changes.
You said out west?
Yeah,
Western part of the state.
He's at App State.
App State, Boone area.
Yeah. So what we're hoping to do is to develop a mountains to sea approach to studying these contaminants.
Right, and that's, it's a slow process, and, we all have other things we have to do, but we're starting to build something. This community of researchers who's working on this, we're starting to get together and talk more about this and get some of this dialogue going. Yeah, Reed.
Just one more question. Regarding these particular organic compounds and being that they're stable, has anybody looked at biomagnification and how that might work its way through an estuarine system or a river system? Has that been a focus at all?
That's been done quite a bit for some of these chemicals. I'm not familiar with every single study, but they have looked at accumulations of foods, starting with water column concentration, phytoplankton concentration, and working their way up into fish species as well. I don't know if I'm not familiar with the avian studies, but they certainly have shown that
in fish, if one of my slides, I can show you later. Yeah, we can have a re-talk.
I'm really glad that you know Laurel Shadler, and just as a comment, one of the things that she is studying, in fact, the main thing that she is studying on Cape Cod is the correlation between the incidence of breast cancer there and the presence of pharma and pesticides in the drinking water. So lest we think that this is pie in the sky stuff and is limited to frogs and fish and young people and old people,
Right.
This is something that could very well be tied to something that's very serious. And I'm really glad that you are in connection with her because she's pretty amazing.
Yeah, she's very good. And actually, it's interesting. She used to work for, her predecessor, whose initials were also LS, Laurel Stanley, used to do the same sort of work. Laurel Stanley and I were also, she and I were actually friends. But it's sort of been interesting that Silent Spring Institute has been really, really proactive about looking at these chemicals from a population perspective and their presence. And I'm jealous. We don't have anything like that in North Carolina that I know of. And a lot of the things I've done in terms of putting together studies are just, here, a couple studies here, a couple studies here. But like I said, it amounts to 20 total. She and then other people have done a lot of work in Cape Cod. And I think a lot of great work is coming out of that group. Yeah.
I think it's partly too because of the environment. The sand on the Cape makes that everything flow into the aquifer, and they're so dependent on several individual aquifers there.
Right.
We have been following them because we're in Plymouth on the largest aquifer in southeastern Massachusetts, just below the surface, and we're all sand. So, all these things quickly, there's not much to stop them from getting into the aquifer.
Yeah.
So do we need to get rid of these septic systems and put in more of their three-stage treatment centers? Is that not a stopgap?
I would say that, so first of all, cost is always an issue. I think having someone go from septic, which I don't know how much it costs to install, but in terms of maintenance, it's pretty darn cheap. But then to move that to do the piping and everything to connect yourself up to a treatment system municipal-wide that has tertiary, it's a cost issue. But something is coming up that they're doing in a lot of coastal areas. Pine Knoll Shores, for example, a lot of the developments down there have what are called package treatment plants. And what package treatment plants are basically they're not tertiary treatment per se, but they're advanced treatment options for developments. So one development will send all their wastewater after it's gone through the septic tank into these package treatment facilities, and they will do some advanced technique beyond secondary, which will then result in the water that's coming out being much better than if it were just a septic system by itself. So Pine Knoll Shores, like I said, there's a whole, in fact, we actually are about to start a study where we're going to look at the efficacy of some of those package treatment systems in dealing with some of these chemicals before and after, comparing it to an area that just has just septic down there. That's supposed to start this summer. Any other questions from anyone?
Well, I'd like to thank CSI for hosting this. I feel very privileged that we were invited to come out and talk to you all. I really appreciate you all coming in, and I appreciate being able to talk to you in such a great facility.
Yeah. Everyone should take advantage of CSI.